Okay, welcome to our second session of Neuro. So my name is Alina Garner, and it's my pleasure to introduce the speakers in this session. And we will start with Maigen Bethea. She is coming from Dr. Methenbegi, coming from the University of Colorado Anschutz Medical Campus. And I will give the floor to you.
Okay, thank you. So good morning, guys. And before I begin, I'd like to thank Kara for the opportunity to be before you all today and present some of my research, and all of the leading edge alum. They've been super vulnerable, super honest, helpful, and supportive. And so thank you all, especially you, Zara. So today I'm going to talk to you a little bit about what I've been doing at the University of Colorado, where I've been focusing on weight loss and sensory neurons. So a continuation a little bit from Cheyenne's talk earlier. We're all in the periphery. So when we ate breakfast this morning, we ingested whatever it was, eggs, and in doing so in our intestines, now we have nutrients, and that in turn cause stretch. And so there are neurons that are along the vagus nerve that are housed in this body known as the nodose ganglia, and they express different markers. And so they basically fall into two types of categories. They can be mechanoreceptors, which sense the stretch or the pressure within the GI tract, or they can be chemoreceptor, which respond to the nutrients and or the hormones that are secreted as food is entering in the gut. But for purposes of today's talk, I'm only going to focus on those receptors that respond and sense stretch within the GI tract. From here, these neurons transmit signals to the brain, or the hindbrain, and my favorite part of the brain is the nucleus of the solitary tract, which is outlined here. And what I have here is a sample immunofluorescent image showing CFOS activation, as that's going to be important for understanding the rest of the data that I show you throughout the talk. But basically, CFOS is going to be a marker of neuronal activation. And so from this point, they send these signals to the brain, and then the brain tells you, hey, you're full, hey, stop eating, and so on. And as you can imagine, if these sensors no longer work, there's nothing telling you to stop eating. And so in the context of obesity, in its simplest form, where it is calories consumed are favored over energy expended, you overeat. And that's not to take away from the complexity of the disease, because we all know that obesity is just more than what you eat. It's a multitude of factors, including genetics and socioeconomic factors. But for purposes of the talk, we're going to go with the most simplistic form, as in calories consumed versus energy expended. And what I hope you can appreciate is in the U.S., the rates are continuously growing in terms of obesity, and they're not getting any better. And if you remember the talks from yesterday from Abby and Kristen, these maps really line well with the incidence of diabetes. And so treatments for obesity fall into three categories. The first being behavioral changes, where you have diet and exercise. And so you can see there's some modest weight loss, but the time is only 4 years. And then you also have rebound. So there have been pharmaceutical therapies. The drug listed here is orlistat, and it's basically a lipase inhibitor. So it prevents absorption of lipids. And again, with that in combination with lifestyle changes, you still see an even more significant reduction in weight loss. However, the long-term effects are only upwards of 4 years. So I'm changing the axis because that's how great of an effect that bariatric surgery has. And so surgical procedures such as bariatric surgery have significant weight loss, and they have sustained maintenance of this weight loss over time. But I do want to mention the caveat that there are a new class of drugs known as glucagon-like peptide 1 receptor antagonists, or GLP-1R agonists, And what they do is mimic the actions of endogenous GLP-1. And so they were used for diabetic therapies to increase insulin secretion. However, it's now become trendy to use these meds for weight loss, and you might know them by the name Ozempic. And so why do you even care about bariatric surgery now? We've cured obesity. And the point is, I'm not saying that everyone needs to have surgery and be cut open to lose weight. Like, that's not by any means what I'm advocating for. But what we should know is that the patents for GLP-1R agonists don't expire until like another 8 to 10 years. So these companies that currently make it, they're the only ones that produce it. And now it's treating 2 severe diseases. So type 2 diabetes and obesity. And so there are not any generic forms available. So it's still the standard or the gold treatment for type 2 or obesity treatment. And again, it's an invasive surgery, but we can learn from the circuits that are altered after VSG to make more targeted therapeutics. And so there are a lot of types of bariatric surgery. However, in the US, the most common forms are sleeve gastrectomy and Roux-en-Y bypass. So vertical sleeve gastrectomy is a process by which you remove 80% of the stomach along the greater curvature. Roux-en-Y is where you literally do a intestinal rearrangement. So you bypass not only the stomach, but the duodenum and connect the pouch directly to the jejunum. But in our lab, we focus on vertical sleeve gastrectomy. And luckily for us, it is like the #1 bariatric surgery procedure in the US. And so, favor was upon us. So we have developed a rodent model of bariatric surgery. And what you see here on the left is just showing that Roux-en-Y in yellow, VSG in gray, like they basically have reductions in weight loss over time, and this is in patients. And what you'll see on the right is that we can mimic these weight loss changes in rats with both VSG and Roux-en-Y bypass. But today, again, I'm only going to focus on VSG. So VSG, aside from the sustained weight loss, you have improved insulin sensitivity, enhanced glycemic control, and changes in feeding behavior. And so I want to focus a little more on the changes we see in feeding behavior. So what we see in these individuals and patients is a little harder because you can't really control what people eat, but you can what mice eat. They're the most compliant patient, right? So what we see is that cumulative food intake is not different from our VSG surgicalized animals and the sham counterparts. However, they have smaller meal size and increased frequency. So they eat smaller, more frequent meals. And people think that this is because of the surgery, that it's restrictive in nature. But what I have here is showing, so if you look at the dotted line, which is representative of a sham animal, and the blue line, which is representative, and the purple line, which is representative of VSG, and look at food intake on the axis, what you see is there are 2 phases. So in the first phase, the VSG animals eat significantly less than their sham counterparts. However, in phase 2, they eat just the same amount. But if you look at the weight loss, they don't gain any weight. Now, if you look at the blue line, this is an animal that is food restricted. So in periods where animals are food restricted, what they then do in order to get to their normal weights, they will become hyperphagic to basically supply the body's demand. And we don't see that in VSG. So this suggests that this is not a restrictive surgery and that something else is happening. So that made us ask, is VSG altering the gut-brain axis? And so we looked in the area that I mentioned earlier, the nucleus of the solitary tract. And so it's the area of the brain that is critical for integrating these peripheral cues. And we quantified CFOS activation or CFOS expression to see what neurons were activated in the context of oral gavage of sucrose, so a nutrient gavage. And we see that there is increased CFOS expression, suggesting that there is something special about the gut that is sending signals to the brain to activate. So we hypothesized that obesity dampens the mechanoreceptor vagal afferent signaling while VSG restores it. And so recently a paper was published that used non-nutritive substances, methylcellulose, to induce stomach stretch, mannitol to induce intestinal stretch, and found that if you induce intestinal stretch, if you look at food intake on the y-axis, you see that it's significantly reduced when you stretch the intestine. And so the long shifting paradigm has been that it's stomach stretch that initiates like reductions in food intake. And so we wanted to mimic this study, and what we did was repeat these studies. So we fasted the animals, creating a biological consequence where the animal would want to eat. As soon as it saw food, we gave them an oral gavage of either the stomach-inducing or the intestine stretch-inducing substance, and then we gave them access to food and immediately measured. So what we see is that we're able to repeat the results. And so if we stretch the stomach here in pink, we don't really see that much of an effect, but when we stretch the intestine, we see a robust effect in reduction in food intake. However, when we feed these animals a high-fat diet for 8 weeks, that effect is lost, suggesting that high-fat diet is altering this intestinal stretch-induced inhibition of food intake. So next, we wanted to see if it's a weight loss specific thing. So we put animals on high-fat diet for 8 weeks and then switched them to a chow diet. So this is weight loss via dietary intervention. We were able to confirm that these animals lost weight, and it wasn't due to reductions in lean mass, only loss of fat mass. And we see that intestinal induced food inhibition is restored with weight loss. We then did weight loss via surgical intervention, where we gave them high-fat diet for 8 weeks and then performed vertical sleeve gastrectomy. Again, we see that over time, weight loss is significantly reduced in these animals. This weight loss is due to losses in fat mass, not lean. And then we see it robustly restores that intestinal stretch-induced inhibition in food intake. And so future directions. So for all of these cohorts, I still have brains that I need to quantify, like over 100. And so I want to see what's happening in the nucleus of the solitary tract for all of these different scenarios and determine the activation in this area of the brain. I also want to assess plasma hormone expression. So these are non-nutritive substances. So in theory, they shouldn't activate any nutrient cascade with hormones such as insulin. And so I want to make sure that is exactly the case. And then what I also would like to do is do duodenal catheter experiments. So bypassing gavage and going straight into the duodenal catheter to see if these effects still are ongoing, to see if it still activates the NTS. And then I also want to use oxytocin receptor Crete mice, shout out Monique, to ablate the intestinal mechanoreceptors. So what I didn't mention is that the intestinal mechanoreceptors express a marker known as oxytocin receptor, and a paper that used optogenetics, so light, to activate these neurons showed that if you activate them, you can also stimulate reductions in food intake. And so I want to see, are oxytocin receptor vagal neurons needed for the success of VSG? Yeah, did take that out. All right, so summary. So what's faded is what I think is happening, but again, I told you, I didn't section the brain, so I don't know. So this is me speculating. So what I think is happening in the context of chow, we have increased intestinal stretch, which is in turn reducing food intake. And I think that CFOS will be increased in the NTS in the context of high-fat diet. There's no difference in food intake. They're still eating what they want to. Intestinal stretch is not necessarily reduced, but their ability to sense is reduced. And I think that CFOS in the NTS will be reduced as well, although there are other studies that suggest that it's even higher, that these are just dysfunctional. And so I want to see in my hands, is it the same? So with weight loss, I think we'll see somewhat of an increase in the NTS. And this is because they're able to sense stretch and reduce food intake. And then lastly, like I put 2 arrows to show like this is a robust effect. So I'm going to see even more CFOS in the NTS, and this is due to the increases in intestinal stretch, gastric emptying, and food intake. And so for the future Bethea lab, so I am a trained developmental beta cell biologist, and I spent 6 years focusing on transcription factors that impact beta cell development and function for the glucose regulation. And now at Colorado, I'm doing nutrients and stretch to see energy homeostasis with the hopes of developing my own laboratory program where I focus on a brain, pancreas, gut axis to look at the regulation of glucose homeostasis and energy homeostasis. So I tried to stay away from doing the reduce, reuse, recycle and not do a circle, but here we are. And so with that, I'd like to thank my lab collaborators and our funding. Thank you.
Awesome. Questions? Okay. I'm going to throw it. No. Okay.
Oh.
Really, really beautiful talk, Allie from Whitehead Institute. I was wondering about the diet and the response to no longer being able to respond to stress. And is it the same response, for example, with a high carb diet versus a high fat diet?
That we do not know. Not necessarily high carb.
Or just even other types of diet?
So again, we have not assessed. I haven't seen anything in the literature, but I know I do have a lab mate who has a high fructose diet, and I wanted to carry out those studies, but because I fast so long, it interferes with liver gluconeogenesis, and that's her main primary outcome. So I hope to assess different diets to see the impact on mechanical vagal.
Really, really cool.
Mechanical symptoms.
That was awesome. This is totally not anything I know anything about, so pardon if this is not a good question, but can you take out parts of the intestine and ask, like, are there parts that are more highly innervated, and can you, like, chunk out the top third, the middle third? I don't know, does it matter?
Yeah, so I have data where I've done that, but I didn't put it here because it's secret.
Okay.
But
so it was a good question.
But
okay.
yeah, you can do that, and I've done similar experiments with the stomach as well, just to kind of hone in on what parts of the intestine and what parts of the stomach are like driving these effects.
Okay. Do you like tequila? I'll get you a drink later. Maybe you can tell me the secret. No? Okay.
I'll tell you the secret.
Okay.
Off camera.
Yeah, it's okay.
I have a similar question because your schematic kind of only shows the innervations to the intestines, but your stretching experiments were also with the stomach. So are there just fewer innervations to the stomach? Is the stomach, like, I don't know, more stretchable? I don't know anything about the system.
So you may not have caught it, but I made like a little blurb about like the classical pathway or cue for satiety has been the stomach. Mm-hmm. But this new paper switched it to the intestine. So
I see.
It's just that the method for which I'm inducing stomach and or intestinal stretch is not to the same extent of those previous studies, which were using intragastric balloons. And so I don't know if the stretch is as stretchy.
Okay. Interesting. Cool. Thank you.
Hi, Maigen. Nice talk. I have a question about, it might be out of your topic or interest. I was wondering, like, because I have read some papers like where they link the role of gut microbiome with the weight loss or obesity. So I was wondering, like, do you have any plan in the future, like, to see any influence of the gut microbiome in weight loss, and also maybe the manipulating the gut microbiome might have some influence on the efficacy of your treatments, like surgical or pharmacological. So have you thought about that?
Yeah, I thought about it. I think I was telling some of the other fellows that I want to say it was Brittany. Yeah, like, it's messy. The microbiome is messy, and then it's just too much variability. And it just seems like a really big feat, a really big task, one that I don't want to take on. But there are others in the field that are trying to assess the changes in gut microbiome. And again, that data is just super murky and muddy because of all the differences with institutions and facilities and diets. So, yeah.
I had one quick question. So this is Abby. For some humans go through the surgery and actually gain the weight later. Do you know if people have followed up to see whether the structure of the diet, so when we go to human diet, does having fibrous food versus soft food affect intestinal stretch? And do you think that's correlated with people who end up going to gain weight, or is it just calories that?
I think food composition does play a part, but again, I think those type of studies in humans would be a little difficult. I should also mention that 80% of all VSG patients are women, and there's a multitude of reasons for that. Like, men are less likely to care about, being bigger. Like, people just say it's vanity for women. And so a lot of the studies have been done in women, and I also have some top secret data that suggests how you lose weight is important as to, like, the restoration in this. Like, it doesn't fare well amongst or across the sexes. But, yeah.
Cool.
Cool.
I have a quick question too. So anyway,
Okay.
I always also use CFOS as an activity marker all the time. It's fantastic. But it's also probably doing something with plasticity. So have you thought about why you would have an upregulation of a factor that is involved in neuroplasticity in the NTS?
No, I have not. And so another thing we're going to do as like a second layer of control is do trapping. Cool. So FOS trap, just to ensure that, for one, this data replicates, and then for two, to figure out what are the neurons that are being activated, and maybe get some more insight into that question specifically.
Cool. Thank you. Okay.